Hey, my name is Zohar, and I would like to present our project about detection of RNA editing sites by GPT fine-tuning. A-to-I RNA editing is a biological process where adenosine, one of the building blocks in RNA, is converted into inosine by ADAR enzymes. So this change is crucial because in the cell, inosine is read as guanosines, effectively altering the RNA sequence, and the protein is called for. So this type of editing allows the cell to diversify their RNA and control gene expression more flexibly. A-to-I editing mostly occurs in the region of double-stranded RNA, where a single RNA strand falls back on itself, creating paired regions that form a double-stranded structure. So predicting the exact site where ADAR enzyme will edit RNA is a complex problem, particularly because ADAR's activity depends not only on the RNA sequence but also on its secondary structure, the way the RNA falls back on itself to create double-stranded regions. So to address the challenge of accurately predicting RNA editing sites, we choose to leverage a fine-tuned GPT model. GPT models are powerful because they are designed to decode complex patterns in data, which is essential for capturing the intricate sequence and structural dependencies that influences ADAR editing. So one of the key advantages of GPT model is their flexibility. They can handle different tasks from generating text to making prediction. For our project, we fine-tuned the model specifically on RNA sequence and structure, adapting it to recognize patterns relevant to RNA editing. Fine-tuning allows the model to achieve high accuracy by focusing on our specific task, which improves its ability to detect editing sites more reliably than traditional methods. So we selected a VN RNA format to represent the secondary structure, as it's effective for GPT model training. These structures makes it possible for the model to learn sequence and structural patterns, allowing it to predict potential editing sites with improved accuracy. So our approach includes two key methods, generation and classification. First, in generation approach, GPT predicts the sequence post-editing, marking likely edited sites. Given the limited data, we use data augmentation, modifying sequences in non-editing region while preserving structure to increase diversity in the training sets and improve the model generalization. So our second method was classification that predicts whether a specific adenosine within a given RNA sequence context will be edited by ADAR. The input of the model is the RNA sequence and its secondary structure, represented in the Vienna format, with the highlighted windows around the potential editing sites. The output is a binary prediction indicating whether editing occurs at these sites, yes or no. In our work, we use two types of context, partial and full. Partial context, which focuses on a local region around editing sites, and full context, which include the entire RNA secondary structure. Using both approaches allow us to explore how much surrounding information is needed to accurately predict editing sites. So to examine the effectiveness of our methods, we focus on A-to-I editing sites within ALO element. These elements are repetitive DNA sequences that are very common in the human genome. Almost all A-to-I editing sites are found within these elements. They frequently form double-stranded structure, the ADAR enzyme act, making them ideal for studying RNA editing and testing our model's predictive power. So an important application of this work is site-directed RNA editing, which allows precise targeting of adenosine bases for editing. This precision is achieved by designing guide RNA that hybridizes the specific sequence, forming double-stranded RNA structure that recruit ADAR enzyme to edit the desired site. By understanding how ADAR enzymes operate, we aim to advance RNA-based therapeutic strategies, such as gene correction techniques, which could provide highly targeted and efficient treatment for genetic disorder. So now looking at the model performance, we evaluated both generation and classification models. So to achieve this performance, we optimized the model classification threshold based on token log probabilities. This optimization allows us to strike a balance between precision and recalls, maximizing accuracy in detecting editing sites both in full and partial context setting. Our classification models achieve a remarkable F1 score of 71% in predicting editing sites, outperforming previous non-AI based methods, and we hope to improve it in the future. So, our research showcases the utility of GPT models in addressing the challenge of RNA editing prediction. By implementing innovative methodologies, including data augmentation and threshold optimization, we achieve meaningful advancement for future work in therapeutic RNA editing. Thank you for listening.